Assalamu alaikum everyone, I am Mohammad Zafar Khan and today I will be talking about the pituitary gland. In today's lecture, I will be talking about the anterior and posterior pituitary glands along with the hypothalamic control of the hormonal secretion. Now the pituitary gland is a small gland also called as the hypophysis which is 1 centimeter in diameter and about 0.5 to 1 gram in weight. It lies in the physiological bone cavity of the base of the brain and is connected to the hypothalamus by a hypophyseal stalk. Here is the hypothalamus and this area is known as the hypophyseal stalk, also known as the pituitary stalk. Now the pituitary gland is divided into two parts or two lobes, the anterior pituitary lobe and the posterior pituitary lobe. This is the anterior pituitary lobe and this is the posterior pituitary lobe. And between these two lobes we have the pars intermedium, which is much less developed in humans but is far more developed in some other animals. Now the embryological basis of both of these parts of the pituitary gland is anterior and posterior pituitary gland is different. For anterior pituitary gland or anterior pituitary lobe we have the red case pouch. For the posterior pituitary lobe we have the neuronal tissue outgrowth of the hypothalamus itself. Now since they have the different embryological basis, they have different types of cells making them, and that's why they have the different type of the hormonal secretions. So let us first consider the hormones of the anterior pituitary gland. These include 6 peptide and various less important hormones, along with the 5 major classes of the cell types. This table includes all of the cell types along with their hormones. We mainly have the somatotrophs, corticotrophs, thyrotrophs, gonadotrophs, and the lectotrophs. Now most of the anterior pituitary gland is made by the somatotrophs, which make almost the 30 to 40% of the anterior pituitary gland, 20% by the corticotrophs, whereas all other types of the remaining 3 types of the cell, they make almost 2 to 5% the total anterior pituitary gland. Now moving on to the hypothalamic control. Hypothalamic control for both anterior and posterior pituitary are different. In the case of posterior pituitary, we have the nervous control. What does that mean? It means that the secretion from the posterior pituitary gland is controlled by the nerve signal that originate in hypothalamus and terminate in the posterior pituitary gland. Whereas in anterior pituitary gland we have certain chemical known as the hypothalamic releasing or inhibitory hormones. That is, these factors or these chemical substances are secreted within the hypothalamus, then conducted to the anterior pituitary gland via the hypothalamus hypophysis portal blood circulation. Now in order to explain this portal blood circulation, have a look at this diagram. In this diagram we have the two types of the capillary network, one lying over here and other in the anterior pituitary gland. Now what happens is that the certain nerve cells in the hypothalamus, they secrete the hypothalamus releasing or inhibitory factors into first this part of the capillary network, then this capillary network combines to form another capillary network in the anterior pituitary gland where these chemical substances are released. And in the anterior pituitary gland, these chemical substances act on the glandular cells which further control the secretion of the pituitary hormones. The anterior pituitary is a highly vascular gland with extensive capillary sinuses among the glandular cells. All the blood that enters the sinuses first passes through another capillary bed in the lower hypothalamus. The blood then flows through the small hypothalamic hypophyseal portal blood vessel, that is these ones, in this direction, to the anterior pituitary sinuses. The lowermost portion of the hypothalamus is called as the medial eminence. This part, this is the medial eminence, which connects the inferiorly to the pituitary stalk. Small arteries which penetrate the medial eminence and then to the additional small vessels return to its surface, which together forms the hypothalamic hypophyseal portal blood vessels. Now the hypothalamic releasing or inhibitory hormones are secreted by the special nerve fibers in the medial eminence. That is, these fibers are located over here in this area. The neuronal cell bodies that give rise to the medial eminence are located in a discrete area of the hypothalamus or the basal brain. The specific location of these cells is not known, so it will be confusing to talk about them right now. As said earlier that the hypothalamic releasing or inhibitory hormones control the secretion of the further hormones. These hormones are given in the table. This table is also taken from the Guyton. It is the table 76.2. In this table you can see that there are various hormones or releasing factors which control the further secretion of the anterior pituitary hormones. Note that the anterior pituitary hormone further releases the hormones which will act on the target cell and influence their secretions. Now let us talk about the pituitary gland. The pituitary gland is composed of the glial cells called as the pituocytes. The pituocytes do not produce any hormones but are the supporting cells of the bulbous-like endings of the nerve fibers which secretes their hormone here. The hormones come from the supraoptic and the paraventricular nuclei located in the hypothalamus. These hormones include antidiuretic hormone and oxytocin. Antidiuretic hormone is most commonly produced in the supraoptic nuclei, whereas the oxytocin is produced primarily in the paraventricular nuclei. But both of these nuclei can produce 10% of the other hormones. That is, the supraoptic can produce 10% of oxytocin and paraventricular can produce 10% of the ADH hormones. Let us talk about the physiological function of the antidiuretic hormone. Now we are all well aware of that the antidiuretic hormone produces the concentrated urine. This phenomenon takes place in the following way. Antidiuretic hormone increases the water reabsorption from the collecting ducts. In the presence of the ADH, the water reabsorption from the collecting tubules increases by many folds, thereby producing a very concentrated form of the urine. Now the process by which the ADH increases the water reabsorption can be explained in the following way. When ADH acts on the cell, it first combines with the membrane receptor, then activates the adenylyl cyclase. This adenylyl cyclase will then further activate the cyclic AMP, which will then in the tubular cell cytoplasm produce certain proteins. These are the aquaporins. These aquaporins are basically the water permeable proteins. They allow the most of the water to be reabsorbed into the tubular epithelium, tubular cells interstitium, from where the water will be transported to the collecting ducts via osmosis. In this way, in the presence of ADH, this whole process takes place in about 5 to 10 minutes. And in the absence of ADH, this whole process will be reversed in 5 to 10 minutes. In this way, ADH will increase the water reabsorption from the kidney's epithelium. Now let us talk about the regulation of the ADH. Whenever the extracellular fluid osmolarity is increased, ADH will also increase. This can be explained on the basis of a special type of the receptors located somewhere near the hypothalamus called as the osmoreceptors. Whenever the fluid concentration in extracellular fluid will increase, water will move from the osmoreceptors to outside via osmosis, which will reduce the size of the osmoreceptors. Osmoreceptor will then generate the signals. The signals will tell the hypothalamus to increase the ADH secretion. In this way, the osmoreceptor regulate the ADH. Reverse will take place in the absence of a high solute concentration in the interstitium or extracellular fluid. Now we already know the effect of ADH on the low blood volume and the blood pressure will stimulate the ADH secretion. Let us briefly discuss the situation. Whenever the blood volume or blood pressure is low, it will send a signal to the hypothalamus to release the ADH. ADH in turn will retain water and will cause the vasoconstriction, which gives it to other name of the ADH, that is the vasopressin, that is the muscular constrictor. Now whenever the vascular constriction will occur due to the increased blood volume and increased arterial pressure, it will produce the stretch. either on the artery of the heart or onto the baroreceptor. This stretch will cause the suppression of ADH release. That is, whenever the blood volume falls, ADH concentration will rise, and whenever the ADH concentration has arrived high enough to retain water and cause vasoconstriction, a stretch will be produced which will decrease the ADH secretion. In this way, the ADH secretion is regulated by a negative feedback mechanism via low blood volume and blood pressure. Now we have the second hormone of the pituitary gland, which is the oxytocin. Now oxytocin has the two primary functions. One of them is the contraction of the pregnant uterus, whereas the second important role of the oxytocin is the milk injection. The milk injection basically takes place whenever the suckling stimulus is applied by the infant, which will then stimulate the nerve, and the signal will be sent to the hypothalamus. In the hypothalamus, the paraventricular nucleus and somewhat supraoptic nucleus will be activated, and they will release the oxytocin. Oxytocin then acts on the myoepithelial cells, which are located outside of and from the lattice network surrounding the alveoli of the mammary glands, which will then will cause the ejection of milk. So in this way, oxytocin increases the milk production during the suckling stimulus applied by the infant. So this was all for today's lecture. If you have any queries, feel free to comment them, and God bless you all. Thank you.